Hello everyone, welcome to today's live broadcast, Advances in Live Cell Imaging. I am Bob Woodard of LabRoots and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Leica Microsystems. Leica Microsystems is a world leader in providing innovative microscopy, camera, and software solutions for imaging and analysis of macro, micro, and nanostructures. For more information, please visit www.leica-microsystems.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to participate during the event. You can submit questions and comments by typing them into the Q&A box, which can be found by clicking on the green Q&A button in the lower left of the presentation window. We'll try to answer as many questions as we can. Also, you can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. Finally, if you have any technical problems viewing or hearing this event, please click on the support button at the top right of the presentation window or submit your questions using the Q&A button. I would now like to introduce today's speakers. Dr. Johathan Jones is Director and Professor at the School of Molecular Biosciences, Washington State University, and Dr. Jennifer Horner, who is the Global Product Manager, Life Science Division of Leica Microsystems. Please join me in welcoming Dr. Jones and Dr. Horner to today's event. I will now turn the presentation over to them.
I wish to thank the organizers for asking me to speak today. The topic of my presentation will be the movement of skin cells and how matrix receptors and their associated proteins regulate this essential feature of wound healing and cancer. First, let me thank the individuals in my lab who performed the studies I will be describing today. I wish to especially thank former lab members Kevin Hamill and Christina Kligys, and two current lab members, Susan Hopkinson and Sho Hirayasu. I would also like to acknowledge the agency NIAMS at NIH that has funded our work over many years, and the Dermatology Foundation for funding Kevin Hamill during his stay in my lab over the last few years. The focus of this presentation is on communication between cells and their microenvironment. This is essential for development, homeostasis, and repair of tissues. Our research in this area focuses on those molecules which mediate the interaction between epithelial cells of the skin and the extracellular matrix. The topic of my talk today will center on the role of matrix receptors, which belong to the integrin family, and their associated proteins in the motility of a keratinocyte, that is a skin cell that's found in the epidermal region of the skin. And we are interested in this motility of these cells during wound closure as well as metastasis in cancer. Work in the role of matrix receptors in cell motility has been well studied by numerous groups. Primarily, the focus has been on adhesion structures called focal contacts or focal adhesions and their associated actin cytoskeletal elements. The immunofluorescence image that I took from the front cover of Science magazine shows a single fibroblast cell stained with antibody against a focal adhesion protein in green and actin cytoskeletal elements in red. Notice that the red bundles of actin terminate on green focal contacts. These termination points appear yellow. This is a very simplified view of focal adhesion structure indicating some of its major components. However, I also would like to point out that there are over 100 proteins that have been shown to associate with the focal adhesion. The focal adhesion not only acts to tether the cytoskeleton to the cell surface and tether the cell itself to the extracellular matrix, but is also a hub of signaling from the matrix to the cytoplasm and vice versa. The current notion for how focal adhesions regulate motility is summarized in the following diagram that I took from a paper from 2007. At the leading front of the cell moving over a substrate, a protein called cofilin severs actin filaments comprising the actin cytoskeleton. This results in the formation of new actin filament ends to which fresh protein actin monomers associate. The newly formed polymers then facilitate cell extension and cell protrusion events. Focal adhesions stabilize cell interaction with matrix, and this allows the cell to develop traction forces. These forces pull the back end of the cell forward. Indeed, detachment of the back end of the cell in combination with traction forces and actin polymerization allows the cell to move in a forwards direction. The scheme I just described is complicated in the cells we work on, that is epidermal cells of the skin, since they possess not only these focal adhesion structures that I just mentioned, but an additional matrix attachment device termed a hemidesmosome. So let me spend a few moments showing you the structure and molecular composition of this thing called the hemidesmosome. This schematic is of intact skin showing the multi-layers of keratinocytes in the epidermis. These layers of epidermal cells sit on a connective tissue or dermis composed of extracellular matrix, fibroblasts, immune cells, blood vessels, glands, and nerves. The electron micrograph that has now appeared shows the region where an epidermal cell or keratinocyte interacts with the dermis. The four open arrowheads mark electron-dense structures, and these are the hemidesmosomes of the skin. Because of this electron-dense appearance, hemidesmosomes have been described as spot wells that function in intact tissue to interact the epidermal layers with the connective tissue. And this is consistent with what we know about hemidesmosome function, as I will describe next. This unfortunate baby has a disease called junctional epidermolysis bullosa, or JEB for short. JEB babies lack formed hemidesmosomes, and an electron microscopic view of a cross-section through a blister of one of these poor patients is shown towards the right. You can see an intact layer of skin cells that has detached from the underlying dermis. This is because of the lack of hemidesmosomes, which are not formed, and therefore the epidermal cells do not attach appropriately to the basement membrane and the connective tissue. Simple abrasion of these poor children results in blister formation because of the separation of the epidermal cells from the underlying connective tissue. Let's now look at the molecular composition of the hemidesmosome. In this diagram, I'm showing you all of the known major components of the hemidesmosome. And the first thing I should point out is it is a somewhat less complex structural entity to the focal adhesion that I mentioned before. I'm going to talk about several different protein components of the hemidesmosome during this talk, and I'm going to mention these here. So the plaquin molecules at the top, called BPAG1E and plectin, these are involved in tethering the cytoskeleton to the cell surface. I'll mention BPAG1E a number of times during this talk. I will also focus on the transmembrane components of the hemidesmosome, namely alpha-6 beta-4 integrin and Col XVII. Alpha-6 beta-4 is a heterodimer composed of a single alpha and beta subunit. It is a matrix receptor. Col XVII is an atypical collagen. It's a type 2 membrane protein, that is, it spans the membrane with its end terminal in the cytoplasm and has a large collagen-like extracellular domain. It binds BPAG1E and beta-4 integrin in the cytoplasm, and alpha-6 integrin and a matrix molecule called lamin 332 in the extracellular space. Interestingly, lamin 332 acts as both a ligand for collagen type 17 as well as integrin alpha-6 beta-4. So far, I've emphasized that hemidesmosomes tether cells down. So what happens during skin remodeling that results from wounds or tumorigenesis? So the first thing that happens is to allow cells to move, hemidesmosomes in the undamaged epidermal cells are disassembled. This is well established in the literature. The dogma then suggests that they only reform once cells have closed the wound. In fact, as I diagrammatically represent here, the literature would imply that focal adhesions assembled at the front of the cell, moving over a substrate, predominating mechanisms that facilitate wound closure. But what about hemidesmosomal proteins in motile cells? My object today is to convince you that hemidesmosomal proteins are involved in wound closure in an active fashion by regulating both signaling and structurally by regulating cytoskeletal dynamics. I hope to show you that they're not simply acting to restrict the motility of even a moving skin cell. So what is the experimental evidence for hemidesmosome protein involvement in motility? First, hemidesmosome proteins, as I'll show you in a few minutes, localize to the right place at the right time. Moreover, hemidesmosome protein deficient cells exhibit aberrant motility, and I'll show you that soon. So first of all, let's talk about where hemidesmosomes are in actively migrating cells. So this image, a set of images, is of skin cells that were grown to confluence in vitro. The lawn of cells was then wounded using a narrow pipette tip. The culture was then fed with medium to activate healing of the wound. The culture was then fixed and stained at about 4 to 6 hours after this wounding process. At that time, the cells had actively migrated into the wound created in vitro. The wound site is indicated on the phase image of the cells at the left. On the right, the fixed cells were then stained to show beta-4 integrin localization, which is shown in red. You can see that beta-4 is concentrated at the very front of the cells moving over the wound site. I should also add that the labs of Bill Carter and John Olerud at the University of Washington in Seattle have also observed comparable localization of beta-4 integrin in vivo in human skin wounds undergoing healing. We repeated the same in vitro wound healing assay and stained the migrating cells that were moving into the wound site with beta-4 integrin antibodies in combination with antibodies against another hemidesmosomal protein, one I mentioned already, called BPAG1E. These proteins clearly co-localize at the leading front of cells moving into a wound site. Okay, so what about hemidesmosome protein deficient cells? What's their motility phenomena? So in this study, we used cells derived from a patient lacking beta-4 integrin, one of these 4 patients suffering JEB. These were kindly provided by Peter Marinkovich at Stanford University. The cells were grown to confluence, scratch wounded as I described before, and then healing measured microscopically. At time 0, both wild-type keratinocytes and the keratinocytes derived from the JEB beta-4 integrin deficient patient showed similar wound shapes. By 48 hours, normal cells have clearly covered the wound site in vitro. However, the beta-4 integrin deficient cells have significantly slower in their migration capacity and are slower in healing a scratch wound in vitro. So to ensure that in fact this was due to loss of beta-4 integrin and nothing else that's happened to the cells since they've been cultured, we rescued the cells by infecting them with a virus encoding beta-4 integrin and in fact induced beta-4 integrin in these beta-4 integrin deficient cells. In this graph, I show you that the wound healing closure of wild-type cells, JEB cells, and JEB cells induced to express beta-4 integrin. The beta-4 integrin rescued JEB cells migrating to the wound at the same rate as wild-type cells, indicating that beta-4 integrin is essential for wound healing in these cells. What about other hemidesmosomal proteins? We've also provided in a number of papers experimental evidence that cells deficient in either BPAG1E or collagen 17 are deficient in motility. So let me use the example of BPAG1E first. We used shRNA technology to derive BPAG1E deficient cells. So we cloned out several knockdown lines that had reduced expression of BPAG1E. So what I've boxed here is an immunoblot that was first probed with an antibody to BPAG1E and then an antibody to mTOR, and the extract was of the cells which were expressing the shRNA specific to BPAG1E. The 3 clone lines indicated here all show reduced expression of BPAG1E, and CN2, which I'll show in a number of different examples later on in the talk, show a reduction of about 72 to 74% in expression of BPAG1E when compared with parental cells. Here I show images of parental cells, HEK cells. These are the epidermal cells that are expressing normal levels of BPAG1E, and then clone 2, which is the BPAG1E deficient cell line. The cells were plated onto glass cover slips and then viewed by phase contrast microscopy. The first thing to notice is this difference in size. The parental cells are much bigger and have spread more efficiently onto the substrate. So we then investigated the motility of both parental cells, the epidermal cells that were expressing normal levels of BPAG1E, and knockdown cells, those that were reduced in expression of BPAG1E, by taking images of cells every 2 minutes for a period of either 1 to 2 hours. I will now show 2 videos. The first is of a parental cell, while the second shows 3 clones, the clone 2 BPAG1E knockdown cells. So this is the first video.
This time-lapse movie shows a fan-shaped keratinocyte moving over a glass substrate. The cell has a leading edge composed of a single lamellipodium.
The next slide shows the BPAG1E knockdown cells.
The 3 cells in this time-lapse movie are deficient in the hemidesmosomal protein BPAG1E. The cells move but stay in place. They extend multiple lamellipodia over their entire surfaces.
So going back to the images, the still images of those 2 movies I show, we can actually derive so-called Roci plots where the motility paths of multiple cells in the population are overlaid on each other. So the parental cells move, as I've said, in straight lines, and you can see this in the Roci plot. In sharp contrast, BPAG1E knockdown cells tend to move in place, as was shown in the movie, and do not migrate in straight lines. We've also generated collagen 17 knockdown cells using the same shRNA technology. In this slide, I show Roci plots of the motility trajectories of a wild-type cell population versus the collagen 17 deficient cells. Just like the BPAG1E deficient cells, the collagen 17 deficient cells move in place. During our analyses of wild-type cells, we noticed that those cells that were moving in a single direction, this is sometimes termed in the literature directed or processive motion, have a fan shape. The leading front consists of a very large lamellipodial protrusion and shows you the direction of migration of this particular cell type as we saw in the movie. So fan-shaped cells are one of the 2 major morphological forms that we observe in a population of wild-type cells. The shapes of the 2 forms are shown here by phase contrast microscopy. You can see that a non-fan-shaped cell possesses multiple lamellipodia. We've actually quantified the numbers of cells exhibiting a fan shape versus cells with multiple lamellipodia, and you can see that in this normal cell population, there's a significantly larger number of fan-shaped cells versus non-fan-shaped cells at any one time. We next followed the fate of a fan-shaped normal keratinocyte over time. This series of images of such a cell was made over a 50-minute period. At time 0, the cell is fan-shaped and moving to the left. It encounters an object and by 20 minutes has developed multiple lamellipodia and is no longer fan-shaped. By 40 minutes, one sees that one lamellipodia seems to predominate and the cell resumes a fan shape and begins to move in a straight line back the way it came. So our data indicate that keratinocytes exhibit a fan shape when moving in a directed fashion. Such cells possess a single lamellipodium. When the cell changes direction, each one first stops, develops multiple lamellipodia, and loses its fan shape. At some point, one lamellipodium seems to predominate, the cell then resumes a fan shape, and the keratinocyte moves in a new direction determined by the predominating lamellipodium. Based on our analysis of wild-type cells, we then reviewed the morphologies of some of our hemidesmosome knockdown cells. And in this instance, I'm showing you data of collagen 17 deficient keratinocytes. So what we noticed and what our quantification showed is there is a significant decrease in the number of fan-shaped cells in collagen 17 deficient cell populations. Moreover, we could rescue this defect by forcing expression of a truncated rescue construct encoding a collagen 17 fragment. We also looked at the persistence of the few fan-shaped cells in our knockdown population. And what we notice is that the persistence of the fan-shaped is significantly reduced in this rare fan-shaped knockdown cell. So far, I've just focused on the consequences of hemidesmosome protein knockdown on cell shape and motility. This raises the question as to how does beta-4 integrin and its associated proteins, BPAG1E and collagen type 17, regulate the mechanisms of motility. One clue comes from a paper from the Marinkovic lab. They showed that beta-4 integrin deficient cells exhibit low RAC1 activity. RAC1 is a small GTPase which has long been known to be intimately involved in cell motility mechanisms. Our data also support a role for RAC in mediating the motility effects of beta-4 integrin and its associated proteins. So let me show some of the data that indicates this. This is all published information. In this study, so first of all, we can show that we can rescue RAC activity in the JEB beta-4 integrin deficient cells by inducing expression of beta-4 integrin. And here we're showing a RAC activation assay in both knockdown or the cells that are deficient in beta-4 but JEB cells, as well as JEB cells expressing beta-4 integrin protein. In this study, we took proteins that co-precipitate with beta-4 integrin antibody, processed them for Western blotting, and then subjected the Western blot to Western blotting with antibodies against potentially associated proteins. So as would be predicted, alpha-6 integrin, the partner of beta-4 integrin, is co-precipitated with beta-4 integrin from extracts of normal keratinocytes. But what I want to show you that in addition to alpha-6, RAC1 co-precipitates with beta-4, indicating that alpha-6 beta-4 integrin exists complex with RAC1 in actively migrating keratinocytes. Of course, this leads to the question, okay, what does RAC1 regulate in actively migrating keratinocytes? To make a long story short, in a series of papers first authored by my postdoc, Christina Kligys, we showed that RAC activates the Slingshot phosphatase, and that induces dephosphorylation of cofilin, which induces activation of cofilin, which leads to actin remodeling. So now we can put together a model of what alpha-6 beta-4 integrin is doing in actively migrating keratinocytes. So upon ligation with matrix, alpha-6 beta-4 integrin activates RAC, which then activates cofilin, to induce actin remodeling and protrusion, i.e. lamellipod formation, which is essential for the directed migration of skin cells. However, I should also emphasize that there is no evidence that there is a direct association between beta-4 integrin and RAC. Rather, in a series of publications, our data support that alpha-6 beta-4 integrin mediated signaling requires a scaffolding protein, namely BPAG1E. So what's the evidence for this? Firstly, the ability of beta-4 integrin to bring down RAC is significantly reduced in BPAG1E deficient cells, as I show here and indicate by this asterisk. You can see much reduced RAC1 that precipitates with beta-4 in the deficient cells. Second, RAC activity is significantly decreased relative to controls in BPAG1E knockdown cells. Moreover, cofilin is phosphorylated. That means it's inactive in BPAG1E knockdown cells. Moreover, in data that I haven't got time today to tell you, our data also indicate that collagen 17 is an essential linker that brings BPAG1E to alpha-6 beta-4 integrin complexes in active migrating cells to allow this signaling through RAC to occur. So conclusions for this first part of my presentation are that the plaquin molecules molecule BPAG1E in combination with collagen type 17 and alpha-6 beta-4 are multifunctional. In an intact tissue, they stabilize epidermal dermal adhesion. Without them, you get the blistering disease junctional epidermolysis bullosa. However, BPAG1E, collagen 17, and alpha-6 beta-4 integrin play previously unappreciated roles in migration. Our data imply that the complex of these proteins is required for signaling to RAP1 and cofilin. Such signaling is an important regulator of lamellipodial dynamics, leading to directed migration, an essential aspect of wound healing, and an important mechanism underlying the dissemination of tumor cells. So the next video provides a pictorial representation of what we think is occurring during motility of the skin cell. The video, please.
Following wounding of the skin, cells at the wound margin begin to move into the wound bed. In the leading cells populating the wound, alpha-6 beta-4 integrin messages are likely co-translated, such that their protein components can be targeted to the leading edge of the cell. Alpha-6 beta-4 integrin then interacts with its extracellular ligand, and in the cytoplasm recruits the BP antigens, namely collagen 17 and BPAG1E. BPAG1E is a scaffold for RAC1 and cofilin, which is responsible for localized actin cytoskeleton remodeling. Actin monomers add to the remodeled actin polymers and push the leading edge or lamellipodium forwards. The hemidesmosome protein complexes also stabilize the lamellipodium. This process then repeats to facilitate forward cell movement.
So far, I've presented mostly data that's been already published, but now I want to talk about work we recently submitted for review. The first question we've been asking leads from our model. One speculation in this model is that alpha-6 beta-4 integrin and or the BP antigens, BPAG1E and collagen type 17, may associate with the actin cytoskeleton in moving cells. So we have localization to support this possibility. In this set of images, we show co-localization of localization of a focal adhesion protein, paxillin, which is shown in red, actin, which is shown in white, and collagen type 17 in green, in a fan-shaped control keratinocyte. Look carefully at the high-power view of the three images which have been overlaid on each other. This shows actin bundles at the base of the lamellipodium of a moving cell. One can clearly see collagen type 17 in green aligned along the white actin filaments. Paxillin appears in stripes, perpendicular to the same actin bundles. Of course, this leads to the question, how does collagen 17 show this interaction with actin bundles? So over a decade ago, we showed that collagen 17 binds actinin 4, an actin binding protein, using yeast-2 hybrid assays, but we didn't really understand at the time what that meant for moving keratinocytes. So in this series of images, we demonstrate that actinin 4 co-distributes with collagen 17 along actin bundles at the base of the lamellipodium. This is best shown in the high power. Actinin 4 in red and collagen 17 in green co-distribute along these actin bundles, and because of their co-distribution, they appear yellow in the overlay. In other words, actin 4 likely mediates the interaction of collagen 17 and hence hemidesmosomal protein complexes, including alpha-6 beta-4 integrin with actin. The actin cytoskeleton is quite dynamic in moving cells. Skin cells are no exception. Since we have observed association of collagen 17 with actin, we next assayed their relative dynamics in live cells expressing tagged molecules. This still image shows actin in red and collagen 17 in green. The video that I show next shows a movie of the same cells.
This keratinocyte was induced to express red-tagged actin and green-tagged collagen 17. The cell was then filmed as it moved over its substrate. At the leading edge, one can see extension of very fine fibers of actin into the developing lamellipodium. Actin bundles move towards the back of the lamellipodium, where their movement appears impeded once they contact green puncta of collagen 17 molecules.
So here I'm showing you some stills from that movie that was just shown. In this higher power view, the actin bundles are shown and have been followed for a period of about 12 minutes. What happens is once they hit these complexes of hemidesmosomal proteins, which are in green and which now show up as a box, their movement is impeded. We next compared the dynamics of actin bundles in live normal cells and cells deficient in the hemidesmosomal protein collagen type 17, which we previously described as being associated with actin cytoskeleton. So there's a significantly increase in actin bundle dynamics in the knockdown cells. So we've quantified this increase in dynamics, and it is significant. Moreover, it is actually rescued by expressing the collagen 17 fragment that I've also mentioned before. So therefore, this deep increase in actin dynamics is due to the loss of collagen 17 in these cells. We also assessed the angle between the base of the lamellipodium and the arcs of actin bundles in both control and the collagen 17 knockdown cells. In knockdown cells, the angle is significantly reduced. This is consistent with an increase in bundle dynamics. So to reconcile all of the observations I've just shown you, we've come up with the following model. As a skin cell moves forward, actin bundles generated in the lamellipodium move backwards. This backwards movement is impeded by hemidesmosomal protein complexes, primarily collagen 17. However, in the absence of collagen 17, there is no impeding of the actin bundle backwards movements, and hence actin exhibits enhanced dynamics. The effects of collagen 17 loss on actin dynamics led us next to ask whether collagen 17 is involved in the generation of those forces, traction forces, needed to move a cell forward that I mentioned early on in my talk. Let me explain. In this schematic, the authors have depicted the development of traction forces, forces developed by contraction of the actin cytoskeleton on adhesion sites, in this instance focal adhesions, exert a force on the substrate that allows a cell to move forward. So this schematic shows how traction forces can be evaluated in cells in vitro. To do so, cells are plated onto a pliable gel into which the investigators incorporated fluorescent beads. As a cell adheres to and moves over this gel, the forces it exerts on the gel induce movement in the beads. An image is taken of these cells. Following visualization of the cell on the beads, the cell is removed and an image of the relaxed state of the beads is taken. Then by comparing the beads before and after the cell has been removed, we can generate force maps. This is a quiver plot or a force map of a fan-shaped keratinocyte and depicts the forces this keratinocyte is exerting on its substrate at one time point. Forces are represented by arrows. The colors of these arrows represent the strength of the forces. The cell is moving in the direction of the red arrow. In this cell, forces are concentrated at one flank of the lamellipodium at this one time point. We can generate these force maps not only of individual cells and cells at any one time point, but of epidermal cells moving over this substrate, as I will show you in the next video.
The traction forces generated by a keratinocyte moving over a gel substrate are shown in this video. The cell itself is visualized by differential interference contrast microscopy, DIC. Individual quiver plot images were overlaid on the DIC images and assembled into a video. The arrowheads depict the forces exerted by the cell. Yellow forces are the highest and appear to move from one flank of the cell to the other as it traverses the gel. As it approaches a cell, it begins to turn. Concomitantly, it generates multiple lamellipodia, and traction forces begin to relocalize to these areas.
So these still images show the DIC images of the same cell that was shown in the video. So what one can see is that traction forces move from one flank of the cell to the other, as indicated by these red arrows. In other words, the cell appears to be moving step by step over its substrate, and we depict this in the diagram shown in panel C. We've also compared the traction forces exerted by normal and our collagen 17 hemidesmosomal protein deficient cells. In the deficient cells, traction forces do not appear to move from flank to flank, but rather are randomly displayed at the cell surface. These results lead us to this particular model. I admit it's very highly speculative, but at this point it fits the data I've just described. The model incorporates both the functions of focal adhesions and hemidesmosomal proteins in motility. You will remember that I demonstrated that the focal contact protein paxillin is enriched at the flanks and along the lamellipodium. So let me take you step by step through the model. So in step 1, actin filament bundles and arcs are stabilized by focal adhesions at the flanks of the leading edge. Backward movement of the actin arcs caused by arc contraction is inhibited by hemidesmosomal protein complexes enriched in collagen type 17. Hemidesmosomal protein complex interact with actin and therefore this allows asymmetric arc contraction leading to asymmetric traction forces at any one flank. The traction forces at one flank induce release of cell matrix adhesions at that flank. The cell is then allowed to pivot on the attached flank, and the released flank moves forward. Actin filaments reattach at focal adhesions at the newly positioned flank. This process repeats at the opposing flank, such that the cell moves in a stepwise fashion over its substrate. So to conclude this second part of my talk, I hope I've convinced you that collagen 17 binds to actin, likely through actinin 4 in keratinocytes. Collagen 17, by doing so, is then able to stabilize actin arcs at the base of the lamellipodium. Collagen 17 then regulates directed migration through both scaffolding signaling as well as determining traction force localization in a moving cell. So what are we doing next? Well, to date, we focused on single cell motility. Now, in a wound healing system, cells move as a collective sheet. So we're very interested in the impact of what's happening to the sheet as a whole when we induce disruption or deficiency in these hemidesmosomal proteins. Obviously, there's a major story here because what we would like to do eventually is promote wound closure in vivo using either molecular genetic techniques or drug approaches such that we can enhance hemidesmosomal protein expression to enhance wound closure in difficult to heal wounds such as those that diabetics have. So now we're going to hand over to Jennifer, who's going to discuss LICA products.
So thank you very much for your talk, Dr. Jones. I think it's very interesting to hear your research on wound healing and cell movement. And on that subject, I would like to now give you a little introduction and some specifics about using our Leica DMI-8 inverted microscope for live cell imaging. So I'll start with a little bit of information on the DMI-8 itself. So the DMI-8 is our newest inverted research microscope. And it's designed in a way to be fully modular. So here you can see a few examples of different setups of the DMI-8. And the unique thing about this microscope is that you can mix and match modular and motorized components as well as accessories to really fit your needs and your budget. And at any time you can do upgrades to this as well as your needs change or your research change. So it's a really flexible and powerful tool for your research. Another interesting point of the DMI-8 is that we have kind of reimagined the fluorescence axis of this, and we call this the infinity port. And what this does is you can see here in the schematic in red, you can see the infinity port axis. And this gives you direct access into the fluorescence axis without any other lenses or secondary decks involved, which gives you full flexibility to add any kind of imaging or fluorescence illumination that you might need. And you can have, as you can see, there's also two different access points, so you have flexibility as far as how you configure at this point too. And one of the, for sort of normal fluorescence, what we have that you can add is what we call the external fluorescence axis. And this couples directly to the infinity port. It gives you not only a place to attach a liquid light guide, so for high-end imaging sources, but it also gives you access to the field and the aperture planes, as well as it gives you a place where you can put in things like fast filter wheels for fast live cell imaging. And as you can see here, you can have this located either at the back of the microscope or on either the left or right side. So depending on your configuration or your setup, your space constraints, you have flexibility there. And so once you have this fluorescence axis added, you then have the ability to add on things like fast LED light engines. So when you're doing live cell imaging, often you want to image as fast as possible so that you can catch these events as they happen in your cells in real time. So you need a combination of generally a good light source, but also one that can switch colors very quickly if you're doing multicolor fluorescence, as well as some fast cameras which can capture events as quickly as possible. So along with this infinity port that I just mentioned, you also have multiple access points for cameras, so you can attach a fast camera that you might need to do your imaging as well. So then getting into a little bit more of the specifics of what you might need for live cell imaging. So imaging live cells over a long time can be a very tricky thing. So you want to be sure that you're imaging only what's happening within your cells, and the imaging process itself is not affecting your cells whenever possible. So some of the things you need to think about are maintaining a warm temperature so that your cells don't get affected by temperature changes or changes in the gases, so O2 or CO2 typically have to be regulated. And then you want to have things like live cell objectives, which are dedicated towards the type of imaging you're doing, so cells in media, for instance. And then oftentimes you're doing imaging over a long period of time, over hours or even days. So you want to be sure that your cells stay in focus over time, and we have things like like our AFC unit, which I'll explain in the coming slides, which holds your focus over a long period of time. And then finally, you need a software that can combine everything, control it, and also analyze your data. And for that, we have our LASX software. So seen here, you can see there's on this particular setup of the DMA8, you have a very large cell incubator here, which gives you the ability to control your temperature and and also to manipulate, so you can open doors and you can get into your cells if you need to. But there are a lot of other things you can add for your environmental control. And shown here are just a few of the things you might want to have. So along with the large cell incubator, there are things like small incubators which sit on your stage top, which you can see here on the left. And then you need different controllers, and you can see here various controllers from some of our third-party partners that are actually been integrated into our software, so you can directly control and monitor what's happening in your cellular environment as you're doing your live cell imaging. So you can really be sure that what conditions you think are really what's in your cells. Then if we move away from the environmental control, the next thing you want to think about typically is your imaging itself, so your objectives. And we have several different accessories that can really help maintain the best imaging quality possible. First, you can see on the left-hand side, you see our water immersion microdispenser. So this is for water immersion objectives. And it maintains, so if you're imaging over a long time, and if you have a 37-degree environment, for instance, you can have a lot of evaporation on your objective itself, and this microdispenser makes sure that you don't have that you never, depending on how long you're doing your imaging, that you always have your objective in water as it's supposed to be. The next thing in the middle you can see is our moat core objective. So this is a motorized correction collar, which basically makes sure that you have the right refractive index going on your cells. You have the right correction for depending on whether your experiment changes over time. Maybe if you change the temperature or you change the substrate that's going on in there, the moat core can then be adjusted to maintain the right refractive index there. And then the last thing you see on the right-hand side is a live cell objective. So we have some dedicated objectives specifically designed for live cell imaging. In this case, this is a 63x 1.3 glycerol core objective. And this gives you the best, really nice images under physiological conditions. So it's really designed to be compatible with live cell conditions. So once you have your objectives, then you need to make sure that you have focus. You maintain your focus throughout the length of your experiment. And for that, we have a couple of things to assist you. The first is called our AFC. This stands for Adaptive Focus Control. And this is an LED-based system which keeps your cells in focus even if environmental conditions change. So if you add a drug, let's say, or if you change your media, the adaptive focus control can adjust to those changes. And the second thing is that we have within our LASX software, there's a high-speed autofocus as well. So if you combine the AFC and the autofocus, then you can really reduce your phototoxicity that happens in your cells because you reduce your focus time. And this can help when you want to make sure that your cells are getting the lowest level of light possible while still maintaining good imaging. And then finally, the thing that ties everything together is the software. And for that we have, I've mentioned a few times, our LASX software. And this is the nice thing about this software is that we've really made it workflow oriented. So you can see whenever you're doing setting up your experiment, you have a tab like this, which really walks you through the different points of your analysis, from setting up the microscope itself, setting up your experimental conditions, and then processing your data and all the way through to analysis. And in addition, similar to our DMI-8 microscope, the LASX software is very modular. And you can pick and choose which modules are going to make the most sense for your experiment. So in this case, I'm showing a panel from our environmental control module, which, as you can see here, you can control and monitor the temperature as well as the gases, and you can really make sure that your experiment is running as you would like. And then finally, once you collect your data, you then need to make sense of it. And for that, we have several different advanced analysis packages, which can let you do things from the very simple, such as just cell counting, to very advanced, where you can add a lot of different masks, and you can do things like deconvolution, and really get or cell tracking, different things. So taken together with the microscope and the software, you really can get all the accessories you need to make sure that you're setting yourself up for the best experiment you can when doing live cell imaging. So with that, I just want to again show you our DMI-8 and encourage you all to take a look at our website or talk to your local sales rep and learn more about our inverted microscope and how it can help you with your research. So finally, I just would like to thank everyone for joining our webinar today, and I think we will be very happy to take questions now.
Thank you for that informative presentation. Before we get started on question and answer session, I would like to remind our audience on how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the Q&A green button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question is, do non-skin epithelial cells also express B4 integrin, and is it involved in motility?
Yeah, this is Johathan Jones. I'll answer that question. So a number of workers have looked at beta-4 integrin expression and its role in motility in non-skin cells such as breast epithelial cells and other tumor cells. And the work of Arthur Mercurio, who I believe is currently at the University of Massachusetts, suggested that beta-4 integrin is involved in motility in these cancer cells.
Another question is, are the BP antigens important in other cell types?
So the BP antigens include both collagen 17 and BPAG1E. Collagen 17's role in other cell types and other epithelial tissue types is a little more difficult to ascertain, although it is present in glandular material and has been recognized as being a component of lung epithelial cells. Its role is not so clear in these other cell types. BPAG1E, however, is a member of a family of molecules which are called the plaquins, and there are isoforms of BPAG1E present even in neuronal tissues, and the loss of BPAG1 isoforms in neuronal systems, as shown by Elaine Fuchs's lab, leads to neuronal deficits.
Another question, does the Leica DMI8 come with dark field objective?
So I can take that question, and the answer is yes, definitely. So the DMI8 is capable of all of your standard contrasting methods, so bright field, dark field, polarization, DIC, phase contrast. So yes, dark field is definitely one of the things that you can do with the DMI8, absolutely.
Okay, we have another question here for Dr. Jones. Do you quantify the fluorescence signals you acquire from fluorescence imaging, or use it mainly for localizing the proteins of interest?
That's an interesting question, and we are developing methods to quantify fluorescence signals. But at the moment, we look primarily at where the proteins are. But I think it's very important that as we develop the technology and develop the model systems, that we actually quantify using different methods to specify where and how much the protein is localized and where it localizes. Yes, that's a good question.
We have another question. Were the quiver plots to measure traction forces generated using the Leica software?
I believe so. Yes, I think they were because we did those measurements on the Leica SB8 confocal microscope.
Another question. With which parameters did you obtain the video 1, 2, and 4 on the microscope software?
I need further clarification of that question. I'm not quite sure what that question actually, what parameters, I'm not sure what parameters means, what the questioner means by parameters in that question. I'll have to table that one.
All right, do we have any more questions coming in from our audience?
I think there was a question from a colleague of mine called Jeff Siegel, who I know, and he was asking whether there were a number of filopodia in the movies, or beta-4 and the hemidesmosome proteins in the filopodia. It looked like they were. Yes, we do sometimes see the BP antigens localized over and associated with actin in filopodia. We haven't really looked at that very extensively, and we need to do further analyses to confirm that localization. And it also goes on, Jeff has also asked, is paxillin also in these structures or different structures? We do not see overlap of the BP antigens and paxillin, so the paxillin may be localized, and I can't remember whether we see paxillin in the filopodia, but they're not co-distributed.
All right, let's give our audience a moment to submit some more questions. Right. We have another question in from our audience. There were a number of filopodia in the movies. Are B4 and the hemidesmosome proteins in the filopodia? Oh,
I think we answered that question. I answered that.
oh, you did. Oh, sorry. I see it as marked as answered. Sorry. I see another question that came in. Parameters: magnification, time exposure, time scale events, aperture, and light intensity. If they could clarify the question a little more.
I think the individual is asking what magnification we use to generate the movies. I will have to get back to you as a separate email to get that. We generally take, what I can say is that we generally take movies over 1 to 2 hour time periods with an image taken every 1 to 2 minutes to generate our time-lapse movies. But the time exposure, etc., I'll have to get back to you.
All right. I think that is all for questions today. I would like to ask our speakers if they have any more questions, any more comments that they'd like to give to our audience today. But first, I'd like to thank our sponsor, Leica Microsystems, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through 6 months from the live date. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Speakers, do you have any more comments?
I just had one. I think I saw a question that came through which was asking about what's the difference or what are the advantages of a DMI8 versus an SP8 confocal. And I just quickly wanted to, for those that aren't aware, explain the difference. So the DMI8 is a wide field microscope, and the SP8 is a confocal microscope. So the difference, so when you're using confocal, you're using a pinhole of light to collect your images, and it's actually a system that is built on top of a DMI8. So it's an imaging mode, if you will, that is quite different from wide field. So wide field takes an image of the entire field in a certain plane, whereas confocal takes a very small pinhole of light, and in this way they can, confocals can remove a lot of the out of focus light. The advantage with wide field is often if you're doing live cell imaging, you can image very quickly, and sometimes you can also, you can reduce your phototoxicity there. But to learn more about confocal on wide field, I would encourage you to go to our Leica website, where we have a section called Science Lab, and there you can really learn a lot about the basics of different types of imaging modalities. So that would be a great place to get some more information. And then just finally, I'd like to thank everyone for attending today's webinar.
Thank you.
Thank you very much. And again, we'd like to thank Leica Microsystems, and you will receive an email from LabRoots informing you of the on-demand presentation. Goodbye.
Bye.